Hello, everyone. Welcome to today's live broadcast, applying a multi-omics approach to tumor biomarker discovery case studies. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Affymetrix. Affymetrix technologies enable multiplex and simultaneous analysis of biological systems at the cell, protein, and gene level, facilitating rapid transition of benchtop research into clinical and routine use for human health and wellness. Affymetrix, part of Thermo Fisher Scientific, provides leadership and support, partnering with customers in pharmaceutical diagnostics and biotechnology companies, and leading academic, government, and nonprofit research institutes in their quest to use biology for a better world. To learn more, visit affymetrix.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit the questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can at the end of our presentations today. Additionally, you can enlarge the slide window that you're viewing in by clicking on the screen icon located in the lower right-hand corner of the slide window. And if you have any technical problems viewing or hearing the presentation, please click on the support button located at the top right of your presentation window. Or if you like, you can submit your problem through the green Q&A button, once again located on your lower left of the screen. We are happy to say that this is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speakers, Antonia Sepulveda, Yang Yang, and Tamara Lotan. Dr. Antonia Sepulveda received her MD from University of Lisbon and her PhD degree from Baylor College of Medicine. She is professor of pathology and cell biology, vice chair for translational research, and director of the Division of Gastrointestinal Pathology at Columbia University. I will now turn it over to Dr. Sepulveda for her portion of the presentation. Doctor?
Thank you for the introduction. I'm very pleased to present our studies today. We will report on results on our project that is focused on gene expression alterations in pancreatic cancer, which in short I'll use PDAC throughout the talk today, and precancer lesions. When we did these assays identified by human transcriptome array technology. So there are about 46,000 cases of pancreatic cancer each year, and the 5-year survival for this tumor is pretty dismal, with 14% rather survival for stage 1 tumors, early tumors, and 1% for advanced stage cancers. The detection of pancreatic cancer at surgically resectable stage or as precancer lesions is the most important factor for improving survival rates. In addition, screening and surveillance biomarkers to detect precancer lesions in the early stages of PDAC are really not available and are critically needed. So pancreatic cancer develops through a stepwise progression of histopathologic changes of the duct epithelium from the normal duct, which we can see on the left-hand side of the slide, moving through the early stage of pancreatic intraepithelial neoplasia, or PTEN, and then progressing to increasing grade and eventually invasive adenocarcinoma. Studies have shown that low-grade PTENs were detected in about 16% of normal controls and in 60% of chronic pancreatitis, and were found in 82% of patients with pancreatic cancer. I'm hearing that I might need to turn my mic down a little bit. Apologize for that. Maybe here. Okay, so in general, there's fairly limited data on the rates of progression of preneoplastic lesion PTEN to invasive pancreas cancer, but studies have estimated a 1% probability of a single PTEN lesion to progress to invasive cancer. Given this low rate of progression and the fact that we do not know histologically which cases might progress, molecular biomarkers to predict progression to PDAC are very needed at this point. So at the inside of this study, our working hypotheses were multiple, and first, we hypothesized that differentially expressed genes in PTEN and PDAC as compared to normal duct epithelium may represent biomarkers for the development of malignancy, and therefore they may be good biomarkers for early detection or for risk assessment of progression to pancreatic adenocarcinoma. Our second hypothesis was that differentially expressed genes in PDAC as compared to PTEN and normal duct may represent potential biomarkers for diagnosis of malignancy. And thirdly, altered genes with functional roles in central regulatory pathways may be targets for PDAC therapies. So, in order to profile the ductal epithelium, we decided to use LCM because this would allow us to separate the ductal epithelium from the highly heterogeneous background of pancreas tissue and isolate normal duct epithelium, low-grade PTEN, and invasive pancreatic adenocarcinoma epithelium. So in the slide, we can see on the upper panels the stained sections. These are from frozen tissue stained with crescent violet for representative areas from normal duct, PTEN, and the PDAC. And the lower three panels, we can see the corresponding areas after LCM, demonstrating that we could enrich the specimen to near purity. So today I'd like to report the results of coming from 22 frozen samples, which include 9 PDAC, 5 PTENs, and 8 normal ducts. And importantly, these come from trios of samples that represent concurrent lesions from the same patient. So we had 3 trios of PDAC, PTEN, and normal duct. We had 2 duos of PDAC and PTEN, and 4 duos of PDAC and normal duct from the same patient. The table on the right summarizes the concentration and total amount of RNA from each sample that we used for the HTA assays, and we can see that these are in the low range nanogram level. So this slide summarizes the workflow following the recommended steps in the GeneChip WTP co-reagent kit. This is a 2-day procedure in which we first obtain cDNA and perform amplification of cRNA, and in the second day, amplification and labeling of fragmented single-stranded cDNA is done, followed by rehybridization. We use the human transcriptome 2.0 array, and after hybridization, the arrays were scanned in a gene chip scan, after which QC and RMA normalization were performed with the expression console software. The analysis was then done with the transcriptome analysis console and other approaches, including R packages, and these are still preliminary early data, so we are continuing our analysis. Now, this slide shows on figure A above the relative probe cell intensity before normalization, whereas figure B, the graph on the lower part of the slide, shows a relative probe cell intensity after RMA. This stands for robust multi-array average normalization. So then the normalized data were used in subsequent analysis using the TAC version 3.1. As we can see in the graph, the medians of all samples are within bounds, and the samples that represent the adenocarcinoma tissues are indicated by the arrows here on the slide. Maybe I need to click one more time. Yes. So then we did the principal component analysis, and we can see that it did not show significant batch effect as exemplified in this case with the groups of PDAC and PTEN. And of note, the clustering of PDAC is nicely separated from the PTEN samples in blue, whereas the cancer is in red. And this provides confidence to the purity of our samples and robustness of the data, because these are two different biological steps in the cancer progression. So we also did clustering by Pearson correlation of normalized signals, and in this particular slide, wanted to highlight the batch effect that occurred when we included in the analysis a subgroup of samples that were obtained from formalin-fixed paraffin-embedded tissue. So we had 2 patients with trios of samples, so a total of 6 FFPE samples. And while on separate analysis, FFPE assays performed well, they clearly show a batch effect as compared to the frozen section samples. So for subsequent analysis, we excluded the FFPE samples. And when we did that, as we can see in this slide, now we do not see any obvious batch effects. So we started our analysis using standard default criteria, which included up or down regulation at 2-fold level and ANOVA at significance of p less than 0.05. And in doing so, we identified over 2,000 genes, which you can see indicated on the right side of the summary in this slide. Now, when we started looking at the individual groups, and if you look at the lower part of the slide under number 5, we can see that the most frequent alteration for the comparison of PTEN versus normal duct was upregulation of 433 genes. This is inside the red circle. However, when we then compare tumor versus PTEN or normal, under 3 and 4 also within the red circle, we could see that the most frequent alteration was gene downregulation. So we then changed the full change to 1.5 in our criteria. because we wanted to identify transcripts that were similarly altered in penins and tumor. And so in doing so, we ended up finding 16 increased transcripts that were overlapping in tumor and penin as compared to normal duct. And these are possible candidate biomarkers of neoplastic progression. Of note, among these, there were 40 coding genes that we are further looking into. There were, as expected, based on the initial analysis, a larger number of decreased transcripts that were commonly decreased in tumor and penin, and that's shown in this slide. In the center, in the pink area. So we then did pathway analysis using the TAC software, and these obviously showed several significantly affected pathways. In this slide, we show an example which represents a gastric cancer network pathway, in which we can see that on the left side for penin, the right side for PDAC, there are genes that are similarly upregulated in penin and PDAC, such as S100P, whereas there are other genes that are primarily increased in expression, such as CNPF, KIF20B, and TPX2, that are primarily upregulated in tumor but not in penin. And you can see that looking at the red circles on the right side, the diagram network. In addition to the TAC software, we have also used the assign algorithm for pathway analysis. This algorithm uses a Bayesian approach to assign numeric scores to individual samples that reflect pathway activity. And the differences in pathway activity between groups were then tested with the Kruskal-Willis non-parametric tests. So in this slide, we are showing an example of a pathway, the nuclear receptor pathway, that showed differential expression in penin and PDAC compared to normal. And using the assign algorithm, we can see in the graph in the middle, lower part of the slide, normal penin and cancer, normal duct in green, the penin in dark blue, and the cancer in light blue. And here we can see that overall this pathway is greatly reduced in pancreatic ductal adenocarcinoma as compared to penin and normal at high significance. Using gene signatures derived from cell culture, derived cancer pathways, we can see upregulation of well-known pathways such as RAS, which is a typical pathway that's altered in almost nearly all pancreatic cancers, SRC and MEK pathways, some of these in subsets of PDACs. And also we can see correlation between activation of these pathways, predominantly for the SRC and MEK pathway, which is at the lower right-hand side of the slide. These are additional examples of other differentially expressed pathways. And if we look at the lower panels in the slide, in the middle, for example, it is not surprising that the cell cycle checkpoints are upregulated in PDAC versus penin and normal. Now, in recent studies, integrated analysis of PDAC gene expression and genomic alterations reported 32 recurrently mutated genes in 10 pathways. And importantly, in our analysis, we found a number of these pathways to be altered as well, giving us confidence that our data are robust and are in line with other studies, although there are significant differences in methodology because we used LCM, and in these other studies, bulk tissue was used. So we feel that our data might be more representative of the biological process. So, summarizing our findings, we found that there were over 400 genes upregulated in PTEN compared to normal pancreatic ducts. For tumor versus PTEN or normal duct, the most frequent alteration, however, was gene downregulation. We found 60 genes with increased transcripts overlapping tumor and PTEN tissues as compared to normal ducts, among which there were 40 coding genes, and these are possible candidate biomarkers for assessing neoplastic progression and may be useful for early detection of pancreatic cancer. On pathway analysis, we have shown several pathways with significantly different activity between PDAC, PTEN, and normal duct, including previously reported pathways in PDAC. And these provide opportunities for functional analysis and interrogation of potential targets for therapy. So finally, and most importantly, I want to acknowledge the members of our research team, Elena Komisarova, George Sepulveda, Sarah Wood-Konkarnka, and Miriam Shirazi, as well as the personalized genomic laboratory at Columbia University. And additionally, I'd like to acknowledge that the study received funding from an Affymetrix tumor profiling grant. Thank you for listening.
Thank you, Dr. Sepulveda, for that informative presentation. And now I'd like to introduce Dr. Yang Yang. Dr. Yang Yang received her PhD from Nankai University. She is currently an assistant professor in the Department of Obstetrics, Gynecology, and Reproductive Sciences at the Yale School of Medicine. Dr. Yang Yang, please proceed with your portion of the presentation.
Hi, it's my pleasure to be invited. Just before I start, I can't see my slides on the screen. Can anyone help me? Okay, got it. Thank you very much. So this is actually my first time present my work through LabGroup. It's my first time I can share my research with the researcher from all over the world from my office. So I'm quite excited for this. I want to first, this is a project we started with the support from Affymetrix, and with the tumor profiling grant, we were able to profile 30 ovarian cancer samples, and I'm going to share some of the data. And before I share the data, a little background about ovarian cancer. Ovarian cancer is a very lethal disease, and the survival rate hasn't been improved in the last 20 to 30 years. The main challenge for ovarian cancer treatment and detection is this is a disease we still don't have very good method to detect earlier. And as soon as the patient is diagnosed, they will receive standardized chemotherapies. And after several cycle treatment, patient may become disease-free, but the majority of ovarian cancer patients will develop recurrent disease. The recurrent disease usually are very resistant to all the conventional therapies. So our challenge is how we can treat these, or we can prevent the recurrent disease. So that's the focus of my research. In the past few years, okay, let's see the slide moving. Okay. So one of the reasons why ovarian cancer is so difficult to treat and detect is because ovarian cancer is such a complicated disease. It can develop from multiple origins, and based on the subtype of ovarian cancer, they have very different genetic background and pathological features. So in the past few years, our study, we start to understand not only this, in general, ovarian cancer is many types of disease. Also, sometimes in one patient, one tumor, you can detect very complex population. So the ovarian tumor, the heterogeneity is also one of the problems we have to face when we develop new therapy. In our study, we discover when we isolate a population of ovarian cancer cells, they express a high level of CD44 and MYD88. And when we inject these cancer cells into animal, they can form tumor. And in the tumor form in mice, we can detect both CD44, MYD88 positive population and the negative population. And if we look into their gene expression, the positive population, they express high level of stem cell, cancer stem markers. When they become the negative population, they lost these markers. Moreover, we further characterize the difference between these two populations, not only their morphology, also many of their cellular functions. For example, here, I hope you can appreciate this picture. There's a dramatic difference between these two pictures. So the picture on the left is the cells with CD44, which can form tumor. In matrigel, they can grow into a web-like structure. But when they lose CD44, and they don't have the ability to survive, to form this structure. Of course, we also check their tumor forming ability. Only the cells maintain all the markers, they can form tumor. When they lose marker, they don't form tumor anymore. So is it possible these unique population of ovarian cancer cells, they are the cancer stem cells? Is it possible they are the cells really regulate recurrent and resistant to therapies? We are conducting more studies to further understand all these markers. Of course, other groups studying ovarian cancer, they are using different cell surface markers, CD44 and many other markers. I believe we are looking at different populations in ovarian cancer. This disease is far more complicated than we thought before. And in our therapy development studies, we try to mimic the real tumor by mixing these two different population cells. We label them with different fluorescent protein. Here you can see when we treat this mixture of ovarian cancer cells with conventional therapies, and the stem cell population, they survive, the green cells. Only the red cells, the cells lost stem cell marker, they are killed by conventional therapy. And this is Nobling3A is another compound which targets p53 pathway. You can see this compound is also not very effective at killing all the cancer cells. So during our study, we discovered some new compounds which have the potential to eliminate both cell population. Showing here, this is one of the compound, and it can kill both cell population. Hopefully, in the future, we can use this kind of drug to help patients. So to summarize what I just described, in ovarian cancer, it is, or I would say in many other types of cancer, it's possible the primary tumor was already a mixed cell population. And during the therapy, a lot of cells, they are sensitive to therapy, they are eliminated. But the stem cell, the more resistant cells, they survive, not only survive because they have very unstable genome, they may gain more mutation during the chemo and then develop into many new populations, form a more complex, more resistant tumor. This may explain what happened in many patients after many years, maybe they recur after the dormancy period, and these tumors become more aggressive. So what are the molecular pathways behind this transition, this progression? We're still in the process of understanding the mechanism. One of the goal of our research is to understand what happened during this process. But because now we're really limited by our knowledge, and sometimes it's really difficult to get tissue samples. So now our study starts with getting a piece of primary tumor and then the recurrent tumor. Actually, during this process, there's a lot of things happened. We still have difficulty to capture at the different stages. So with the support from Affymetrix, we were able to use both DNA and the RNA array. So why I chose DNA array OncoScan, there's a reason. So this is a recent paper published on Nature Genetics. They actually this group studied many different types of cancer. And their conclusion is that all these different types of cancer can be divided into two types. One type here, summarizing here, are mutation-driven class. And the second class is copy number alteration type of cancer. Ovarian cancer is right here, actually the top one. So in ovarian cancer, especially the most aggressive type, high-grade serous ovarian cancer, copy number variation, copy number gain and loss are very common, and the frequency happen also very high. And this is why we chose OncoScan to detect the copy number variation in FFPE tissues. And in combination, we also choose human transcriptome array to study the RNA. And hopefully, we can find some correlation between DNA and RNA and find some target or some important pathways to explain this molecular mechanism. And so according to our experience working with the Affymetrix kit, we try to follow the instruction very strictly, but it is really challenging when you work with clinical samples. Sometimes you don't get enough tissue. Sometimes you want it to be very fresh, but the experiment process may get delayed. So this is our experience. We were able to get RNA and the DNA from one sample, 10 slices or 3 pieces of 10 micron slice from parent blocks. Some tumors are really small size tumors, including some of the data I'm going to share are from animal tumor xenografts. Some of the tissue are very old, average, I think 6 years old tissue. Some tissue are from patients from 10, maybe 10 years old. And this is the challenge we have to face because ovarian cancer, as I mentioned, many patients, their disease recur like 6, 7 years later. So when you get the secondary tumor, the primary tumor, paraffin block, it's at least 6, 7 years old. And another reason we chose OncoScan is we really like the software. It's really easy to use. It doesn't take a lot of time to learn how to use it, and there's very comprehensive information. And sometimes it's so easy, you can, from the view of the whole genome, and really zoom in to each gene, and it's directly linked to the function and the publications related to this gene. So it's really facilitate our study. So here I'm showing some examples. As I mentioned, high-grade serous type ovarian cancer is the most aggressive. They have a lot of copy number variation. Here we're showing a patient A and a B. So this patient A on the upper panel here has a lot of copy number gain, copy number loss, and the yellow part label is the LOH. So this is a typical high-grade serous type ovarian cancer patient. Another patient, you can see the lower panel, I'm showing here as an example to compare with. This is also an ovarian cancer patient, but a different type. And you can see the copy number variation are quite different. So by OncoScan, we can distinguish different type of ovarian cancer. And the next example I'm showing here, this is a high-grade serous type ovarian cancer. The same patient, primary tumor and the recurrent tumor at the lower panel. You can see the overview. This is a summary of our OncoScan data. A lot of region, copy number gain in blue and copy number loss in red, and a lot of yellow parts, LOH. When the disease recurs, in general, you see they look very similar. But if you zoom in and look into the details, there's another way to show. This is the same patient data. I put it in a different format. By the way, all these figures are generated from the Nexus, the software. So here you can see they look very similar. You can tell this is from the same patient. But if you look closer, in the primary tumor in this region, there were chromosome 3, there's copy number gain, but it's not detected, it's not in the recurrent tumor. And here, there's a copy number loss, and it's not in the secondary tumor. So actually, there is a shift of the cell population in the tumor, and something has changed. So we did the OncoScan for all the 30 tumor samples, paired samples, and we also used exactly the same paraffin samples from the same sample. We extract RNA, and we run the HTA. And this is a summary of all the 30 samples. And you can see all these regions labeled red or green. They are the significantly differentially expressed genes or transcripts, and how they localize in the genome. So it's a lot of data. We are still in the process of analyzing, understanding the data. Some preliminary analyses are already very interesting. For example, here, when we compare we get all the genes, copy number variations from DNA data. We analyze the pathway. We also did a similar with RNA data. It's a very simple analysis, but if you look into the pathway, there are very important pathway, well-known pathway play important role in cancer, like cell death, cell survival. There are also pathway, at least it's new to me, like neural system development related. This is in the top 5. And in the RNA data, we also found the neurological disease related. This is surprise to me. Ovarian cancer hasn't been related to a neural system related gene or pathway. Is it possible this is something we should look into, or is it an artifact from the test? We are doing more validation to confirm this data. And so as I said, we are trying to understand this disease progression process. And we ask more questions. Is it possible using the technology, using the model we have, we take a primary tumor from the patient, and by using a xenograft model or in vitro model, we treat this tumor and try to select the resistant cancer stem cell population. And understand this population, is it possible we predict what are the pathway going to control this disease progression, and it's going to be the resistant pathway in the resistant disease. So is it possible to predict the resistance? To prove this possibility, we try to use animal model to test it. One experiment we did is in mouse, we inject human ovarian cancer cells, and you can see they are labeled with fluorescent, and they develop into very aggressive tumor if we don't treat them. If we treat with paclitaxel, our mice patient, they become disease-free, but after some time, these tumors recur. If you don't treat them, they will become very aggressive again. What if you treat them and these tumors become resistant? They don't respond anymore like the primary disease. So this is actually very similar to what happened in human patients. And we were able to use this model on different drugs. And here's one of the data we got from the xenograft from mouse model. You can see the early tumor and the late tumor. They look similar. There are big regions of copy number alterations. And there are also some regions, if you zoom in, they are gained when in the much later stage of these tumors. We also detect enrichment of a population of cells with KRAS mutation. When we conduct this similar study with different drugs, including the new compound I mentioned earlier, you can see a very different DNA copy number profile here. And actually the Taxol, which is shown in the animal model and in vitro model, very efficient treatment, and it actually alter the genome the most. You can see all these big regions, and if you zoom in, you will find even more different genes, different copy numbers. And surprisingly, the drug, we are testing the new drug, it caused the genome alteration the least. And we still try to understand how this drug works, why the genome is not changed as much as conventional therapy. So when we put all the genes, which are significantly changed, both at DNA copy number level and the RNA level, we found very interesting pathway, including here, I was surprised, the ovarian cancer signaling related gene and pathways here in the center. And we also find the MMP family related pathway and L6 signaling pathway. L6 is a very important pathway in ovarian cancer. And yeah, and the HIF1 alpha signaling pathway. This is related to the metastasis, especially for ovarian cancer. Metastasis happen in the peritoneum cavity, and a lot of environmental factors are hypoxia related. HIF1 alpha pathway play an important role. And this is the RNA data. We found some pathways are correlated to the DNA copy number data. We found also new pathways like CXCR4 signaling pathways and EGF pathways. So summarize that pathway again, in mouse model, we see similar as human model, as I mentioned, the very important tumor-related pathway, and again, the neural system-related pathway here, and here in the RNA data. So now our lab, we are really interested in validating the data and try to conform these data in a more detailed and functional level. So our vision is in the future, based on our improved knowledge of this disease and new models, new techniques, is it possible we will predict the recurrent disease and even develop or screen for therapies that can target the recurrent disease. So before the recurrent disease happen, we can already have these drugs prepared for the patient. Or one step further, maybe we can use these new drugs to prevent the recurrent disease as a chemoprevention approach. So to summarize, we have used, or we have been using Affymetrix FFPE for OncoScan and HTA array. And now we are using the quantogene multiplex and try to conform some of the genes that are differentially expressed from the array data. In the end, I want to thank Affymetrix, a lot of colleagues. They support our research and give us the technical support and including data analysis. And thank you for this opportunity. Thank my collaborators. I have seen the number of our attendees are increasing. I hope some of my collaborators are also looking at this presentation, and I would like to get your feedback for our research. Thank you.
Thank you for your that informative presentation, Dr. Yang. Next it is my pleasure to introduce Dr. Tamara Lotan. Dr. Tamara Lotan received her MD degree from the University of Chicago Fitzgerald School of Medicine. She is currently an associate professor of pathology and oncology at the Johns Hopkins University School of Medicine. Dr. Lotan, the floor is yours.
Great, thanks very much. Let me get my slides. Excellent. So thanks for this opportunity to present our research. And I'm going to tell you a little bit today about some work we've been doing on integrated genomic profiling of primary prostate cancer, which we hope will eventually be useful both for prognosis and for prediction. So prostate cancer is a clinically and genetically heterogeneous disease, and this has really been a challenge in trying to study it and treat it. And we know that some prostate tumors are quite indolent, will grow very slowly, and the patient will likely die of other causes even though they have prostate cancer. And these tumors really shouldn't be treated. On the other hand, we know that a number of tumors are lethal and require pretty aggressive therapy at the outset. And distinguishing between these two different populations of tumors has been a challenge. We have some morphologic or pathologic criteria that help us to make this distinction, but there's really been an active search for biomarkers, particularly molecular biomarkers, that might help pathologists to distinguish between these lethal and indolent subtypes of prostate cancer. So we've and others have felt for a long time that developing a molecular taxonomy of prostate cancer will be critical to minimizing disease over-treatment on the one hand, and also to realizing the promise of precision medicine for patients with advanced diseases. And a number of large-scale efforts have been have taken place really to characterize the genome of human prostate cancer. Probably the largest effort in primary prostate cancer has been the Cancer Genome Atlas. And at this point we know very well what the common and uncommon molecular subtypes of primary prostate cancer are. But a challenge has been that the efforts such as the Cancer Genome Atlas, although they very carefully molecularly subtype large cohorts of prostate cancer patients, do not generally have large amounts of outcome data or very high quality clinical or pathologic annotations. So it's been very hard to map back these molecular subtypes onto the, onto outcomes data. And so our challenge, or we feel the challenge has really been to make the molecular taxonomy, which we now understand in prostate cancer, clinically relevant, hopefully by integrating much of this multidimensional cancer genomics data with a very highly annotated cohort of clinical pathologic specimens where we have very high quality annotations and can try to understand what the meaning is of these molecular subclasses for patient outcomes. So I guess as Dr. Yang has nicely pointed out, their cancers can sort of be divided between those where the copy number alterations prevail and mutations prevail. Prostate cancer, I guess much like ovarian cancer, is one of the solid tumors in which copy number alterations really prevail over mutations. And here are two actually relatively older studies, some of the earliest studies looking at copy number across different prostate cancer cohorts. On the right side of the screen, you can actually see an advanced cohort of prostate cancer patients with metastatic and lethal disease taken at autopsy. And you can see that the copy number gains and losses, which are denoted in blue and red dots, really prevail over mutations across the gene sequence in this whole exome sequencing study. Mutations are noted in yellow, orange, and green. On the left side of the screen, you could see the typical copy number landscape of primary prostate cancer. So we have typically gains of chromosome 8q, for instance, where MYC is located, losses of chromosome 10q, where PTEN is located, losses of chromosome 13q, where RB1 is located, and often frequent losses on chromosome 17 at the TP53 locus. So we understand this spectrum of copy number alterations, and now I think the challenge is to really integrate that with clinical outcomes data, and then also with RNA expression and protein expression data to develop better prognostic classifiers to apply to prostate cancer biopsy specimens. So we undertook a study to begin to do this kind of work using a cohort of specimens from the Johns Hopkins Brady Urological Institute. So we have a large biospecimen repository here of primary prostate cancer with arguably among the highest quality and certainly most extensive clinical annotations available. And so we were interested now in mapping back these known molecular subtypes of prostate cancer which have been described in efforts such as the TCGA onto our cohort where we have excellent clinical and pathologic annotations to try to understand how these genomic alterations correlate with outcomes. And so we proposed to develop something we would call the Brady Genomics portal, which would be an internal database enabling Hopkins researchers to query the tumor cohorts that we have in-house in terms of their genomic profiles, with both genomic and transcriptomic profiling available. So the cohort we started with is one that's actually been previously published by Ashley Ross, who's a urologist at our institution. The reference is down in the lower right-hand portion of the screen. And this cohort consists of around 350 radical prostatectomy specimens, all formalin fixed and paraffin embedded. They were all performed or removed from the patients between 1992 and 2010, so relatively older cohort. And this means that we have a median of 10 years of clinical follow-up on the majority of the specimens, and the outcome that was queried for was metastasis, which is really considered the gold standard in evaluating prostate cancer outcomes. One other advantage of this cohort is that none of the patients in the cohort received any treatment between the time of radical prostatectomy and the eventual time of detection of metastasis. So it's considered to be a natural history cohort in the sense that patients received only surgical treatment with no adjuvant chemotherapy or radiation, which might make discerning the natural history of those cases difficult. One other advantage of the cohort is our colleague George Netto in pathology here had already regraded and restaged all of the cases in this cohort using the most updated pathologic criteria, and that was reported in a PLOS One paper in 2016. And we went ahead and sampled the dominant nodule and quadruplicate from all these cases for both DNA. Previously, RNA extraction had already been done from the same tumor nodules, and TMAs had also been prepared by Dr. Netto and his team. TMAs meaning tissue microarrays, I should clarify. So a little bit of this work has already been presented. This is actually, I forgot to put the reference in the lower corner, but this is a paper by Michael Johnson at our institution in the Journal of Urology, which was published this year in 2016, looking at the ability to use the RNA profiling from this cohort, which had been completed on the Affymetrix 1.0 ST arrays, to predict for one of the most common genomic rearrangements in prostate cancer, which is a rearrangement in one of the ETS family oncogenes. It's ETS family oncogenes. And the most commonly rearranged gene is the ERG gene. You can see on the left-hand panel that when the ERG gene is rearranged, either by a deletion or translocation, it's then under control of an androgen-regulated promoter, typically, and expression increases dramatically. So we can detect the presence of these underlying genomic rearrangements just by the RNA expression or protein expression of ERG oncogene. And you can see that about half of the cases had underlying rearrangements as evidenced by this increased ERG expression, which is what we expected. Perhaps more importantly, we don't have good antibodies to detect rearrangements of the other ETS family oncogenes, such as ETV1, which is depicted here. And so RNA levels can be used to infer the rearrangement status of these cases. You could see that a subpopulation of cases, less than 10% as expected, had overexpression of ETV1, suggesting an underlying ETV1 genomic rearrangement. And tumors with ERG or ETV1 rearrangements constitute probably the main known molecular subclass of prostate cancers. So we were interested in taking this one step further and looking at the expression of these ETS family genes to make these rearrangement calls in situ in small quantities of tissue. So we used the Affymetrix View RNA and a combination of also the ACD RNA scope technology to do RNA in situ hybridization for the three ETS family genes, ETV1, ETV4, and ETV5, for which we don't have good antibodies for detection. So RNA really remains our only available tool to look at overexpression of these genes. And again, overexpression tightly correlates to rearrangement of the underlying genomic locus and leading to androgen-regulated overexpression of these oncogenes. And so you can see here we have some examples depicted of RISH for ETV5, ETV4, and ETV1. The top row on the left panel depicts cases that were positive. So you can see the red dots indicate positive detection of these RNA molecules in the cancer cells, and you could see intervening benign glands, which are negative and just blue, as we would expect. And the bottom row shows you some cases that were negative. And then the right-hand panel shows a nice correlation between the calls for ETV1 rearrangements that were made based on the RISH, or the RNA in situ hybridization, which was done on these tissue microarrays, compared to the ETV1 expression levels seen by the Affy 1.0 ST array transcriptomic profiling. And you can see that there's a very high correlation. There were only 2 discordant cases in the entire cohort. So we can very nicely classify these cases either by grinding up the tissue and doing transcriptomic profiling, or perhaps even more easily by doing in situ hybridization for these transcripts. In addition, on this cohort, we looked to see whether known genetically validated immunohistochemical biomarkers in prostate cancer were performing as we expected them to. Here we're showing immunohistochemistry for P53, which is fairly sensitive and specific for the presence of an underlying P53 missense mutation, which leads to stabilization of the protein. And you could see that in this cohort, sort of as a sanity check, we could tell that in the right-hand panel that cases with P53 overexpression, which have known mutations in the gene, in the P53 gene, have a worse outcome in terms of a cumulative incidence of metastasis in this cohort, as we would expect. So again, just sort of a check to make sure that everything looks reasonable in terms of a correlation of known genomic biomarkers with outcome in the cohort. So next, what we wanted to do was gather relatively high resolution whole genome copy number analysis or copy number profiling on all of these formalin-fixed, paraffin-embedded prostate cancer primary tumors that were in the current cohort. And so we really needed to find a platform that worked in FFPE samples, which was challenging, and one that required relatively low DNA inputs, and we had relatively older samples. And so we tried out the Affymetrix Oncoscan platform, and we've actually had very nice success with it. We've profiled, I think, almost 50 cases to date, and we've had a relatively low failure rate. In the figure depicted on the slide, you could see a typical copy number profile of a typical case with, for instance, a gain on chromosome 8q including the MYC locus, and then with the arrow showing a very specific and focal loss, a deep deletion of the PTEN, a tumor suppressor gene. And below you can see the B allele plot to get a better sense of some of these gains and losses across the chromosome. So this slide shows a sort of the general landscape of copy number alterations we found by Oncoscan in the first 50 primary prostate tumors that we profiled. It looks very similar to one of the earlier figures I showed you from previous work. And again, the key is not to make some new discoveries in terms of copy number alterations, because these are very well known in prostate cancer, but now to really link them to outcomes, which we can finally do with this cohort. So again, you could see common losses at 8p, gains at 8q, as I mentioned, were common, specific losses at the PTEN locus at 10q, et cetera, in this plot. So it looked pretty similar to what we would expect. As an additional validation of some of the specific copy number calls that were made in the Oncoscan platform, we correlated with genetically validated in situ and sequencing assays that we had performed in the lab on the same cases. So for example, we have an immunohistochemistry assay or an IHC assay for PTEN, and you can see an example of that depicted in the figure on the slide. The right-hand spot shows a tumor area depicted by the letter T with PTEN protein loss, which makes the tumor appear blue, compared to the surrounding benign glands, which are labeled with a B. You could see nice dark brown staining suggesting underlying PTEN protein presence in that tumor. The tumor sample on the left has no loss of PTEN at the protein level, based on the fact that the tumor and the benign glands have fairly similar levels of expression. So it turned out that 96% of the cases that had PTEN copy number loss by Oncoscan profiling showed protein loss by IHC. Importantly, 100% of the cases that had homozygous copy number loss had complete protein loss. And then we also saw that among cases with known TP53 missense mutations, which we knew by a sequencing panel performed on these same samples, 75% of them showed copy number loss, presumably at the other allele of P53 in the same case as we would have been expecting. So this was a nice validation of some of the specific copy number calls on this platform. So finally, we're hard at work now trying to integrate all of the RNA profiling with the DNA copy number profiling data, and then some of the RNA in situ and other immunohistochemistry in situ assays into a database that can be queried by researchers at our institution. So you can see here, this is an instance with the help of Dr. Alex Barris, who's also in the pathology department at Johns Hopkins. We've been working on implementing a very local instance of the C-Bioportal database, which is the database developed at Memorial Sloan Kettering and used to report out most of the TCGA studies. We have a local instance of that same database. You could see with the clinical outcome information annotated, pathologic stage, grade, race of the tumors annotated, and now we've integrated, for the first 50 cases we've done, these copy number profiles into that same database. And we're at work now putting in the RNA expression profiling from the Affy ST arrays, and then also some of the immunohistochemistry profiling. And it's our hope that at that point we can begin to integrate DNA, RNA, and protein data to sort of design the optimal and potentially clinically implementable biomarker panel to predict adverse outcomes in prostate cancer. So once we complete copy number alteration analysis on the remaining samples, and we have about 200 to go at this point, we'll integrate this data and the RNA expression data, as I mentioned, to begin to develop prognostic classifiers. And the idea is then to take these classifiers and validate in prostate tumor biopsy cohorts with outcome data information to begin to try to understand which patients can be safely watched and who have genomic profiles that suggest they need potentially immediate treatment. And I think the ultimate goal, obviously, is to create this kind of clinical classifier, both for risk assessment and then perhaps later in progression of disease for predictive biomarkers that may predict efficacy of specific therapies. So with that, I'll just thank the members of my lab who worked on this, and I mentioned many of our collaborators throughout the talk who have been very helpful, and of course the team at Affymetrix and the Tumor Profiling Grant, which was very helpful to get started with some of this data collection. And with that, I guess we'll take some questions.
Thank you so much for your presentation, Dr. Lotan. Just a note before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many of the questions as possible, and so let's just go ahead and hop in and see what we have today. The first question I have is for Dr. Lotan, and it reads, what are the clinical contexts in which copy number alteration profiling could be useful in prostate cancer management?
Yes, so that's a good question. So I think there are a number of contexts in which we could use this kind of technology. One is in the context of managing patients who are potentially candidates for active surveillance in prostate cancer. So these are patients with relatively low burden of disease on prostate biopsy, low pathologic grade of disease. And while I don't think it's likely that we will ever have enough tumor sampled in those cases to run, a whole genome copy profiling test, I think we can begin to take some of the findings from our whole genome copy number profiling to implement very specific copy number alteration assays to query either in situ in the form of FISH or other types of assays in the biopsy sample itself in small numbers of tumor glands. We do know that the burden of whole genome copy number alterations is highly correlated in prostate cancer with adverse outcomes. So it would be nice, I think, in the future if we had some ability to do more comprehensive copy number profiling on these very, very small tumors. But right now, that's challenging. I think a second context of use is potentially in the more advanced setting, perhaps a patient who's already undergone radical prostatectomy and perhaps had recurrence after radical prostatectomy. There we have much more tumor to assay for copy number alterations, so that if we have a good sense of specific copy number alterations that are associated with adverse outcome or even hopefully specifically associated with response to particular therapies, then you can envision a situation in which we could clinically do whole genome copy number profiling in FFPE material from the radical prostatectomy to get some sense of whether the patient is likely to respond down the road to more targeted therapies, or potentially do a similar type of test on the metastatic sample. So I think those are a couple of contexts where it could be useful clinically.
Excellent. Thank you for that answer. Dr. Yang, we have a question for you. It reads and asks, is it sometimes difficult to get good RNA and DNA from FFPE tissue samples? how to improve the quality and quantity of RNA and DNA from FFPE.
So according to our experience, it is indeed to get a good amount of RNA and DNA and also the quality. So first for the array, Affymetric RNA and DNA array, they are very sensitive. They can do really well with very small amount of DNA and RNA. So to improve the quality and quantity, I think one thing actually the most important is if it's possible, when you collect the sample, the tumor sample, when the tumor sample is still fresh, and put it in formaldehyde as soon as possible, as early as you can, because leaving the tissue in PBS for a long time is not good for later the RNA DNA process. And after, if you already have the FFPE sample, what you can do to improve the quality, and there are some critical steps. For example, and to reduce the time your sample slides, the cut slides exposed to the air, in the air, reduce that time. Also, when you extract RNA, the temperature really important. And if there's an incubation time, a high temperature incubation, don't extend that incubation. Longer high temperature incubation will make the RNA and DNA sample into small fragments. That's not very good either. So yeah, a lot of small details, really stick to the protocols and pay attention to the small details. I think it helps a lot for our project. Thank you.
Thank you, Dr. Yang Yang. And since we are running short on time, we only have time for one more question. And Dr. Sepulveda, it will be for you. The question asks, why did you choose transcriptome analysis with the array platform as compared to RNA sequence?
Thank you for that question. It's quite pertinent. So we were interested in characterizing the expression of, for example, the pancreatic duct, which is a very small subpopulation within the large background of multiple cellular components in the background pancreas. And in order to do so, we would need to do laser capture microdissection. So typically, with laser capture microdissection, we obtain very limited amounts of genomic material, namely RNA. So with that, then the consideration is what platform to use if you have limited amounts of material, as well as potentially the quality, because we want to, on validation studies, we want to use larger cohorts and expand our studies of paraffin-fixed tissues. And in those cases, specifically from paraffin, it would not be feasible to use platforms like RNA-seq, for example. So in upfront, using a platform that is not very sensitive to the quality of RNA, and can perform quite well even when the RNA is significantly degraded, we decided that this was a good approach because it allows us to address the gene expression in the fresh tissue, and later, when expanding our studies, also to do the same analysis in the paraffin tissue, and therefore we'll be able to correlate our findings in this early phase as well as with the data that we plan to obtain later.
Okay, well, thank you so much, doctors, for presenting, for your presentations. I would also like to thank our sponsor, Affymetrix, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through February 2017. You will receive an email from LabRoots alerting you when the webcast is available for replay. We encourage you even to share that announcement with your colleagues who may have missed today's live event. We at LabRoots thank you once again for attending, and we'll see you next time. Goodbye.